All right, thank you everyone for joining us both here in Zoom and on YouTube. This is the EM Connectomics MICrONS Explorer tutorial. We're going to go ahead and get started. So as we are going through the program today, please do share any questions that you have for our speakers using the Q&A button here in Zoom, or if you're joining us on YouTube, you can use the YouTube chat. So I'm going to go ahead and kick us off with a little bit of an introduction. All right, so again, this is the Open for Neuroscience tutorial on the MICrONS Explorer, our EM Connectomics data and data analysis tools. We're going to start with just a brief introduction and then move to a demonstration of neuroglancer, software that you can use to visualize this data. And then we have 3 use case demonstrations from Casey, Layla, and Sharmi. We have plenty of time for questions, and they'll be taking questions in the Q&A the entire time. We'll save most of those for the end, but please do post them throughout the talk. So that's my first point. Please put those questions in the Q&A. If you're joining us on YouTube, go ahead and place them in the YouTube chat. And use the Zoom chat to talk to each other about your research interests, but please don't put questions in the chat or they're likely to scroll away and disappear. And make sure you're sending your chat messages to all panelists and attendees so everyone can see your message. And you should be seeing live captions available in Zoom, and that'll be on the recording on YouTube as well. Our panelists today, Sharmi, Casey, and Layla, were all involved in the development of the MICrONS Explorer and its analysis tools. They're going to tell you more about their specific work on this project as they demonstrate how you can use it in your work. In case you missed the symposium today, the Allen Institute is a biological sciences nonprofit located in Seattle with focused research areas not only in neuroscience, but also in cell biology and immunology. We also support cutting-edge research across the world with the Paul G. Allen Frontiers Group. Across the institutes, our scientific process is focused on tackling complex, broad, and hard problems in the fundamental biological sciences. And this approach is guided by 3 core principles: big team and open science, which I'll return to in a moment. And the work generates data, knowledge, and analysis tools, all of which we share publicly for you to use in your work. And to date, we've collected about 14 petabytes of data, which is 2.8 million DVDs worth. And the EM data set that we're about to dive into is the single largest data set of all of them. So the most relevant of our principles today is open science. All of our data and tools are available for you to use in your work. Most of our brain science resources live on brain-map.org, and you can find the link to the MICrONS Explorer, which is at MICrONS-Explorer from BrainMap as well. And in addition to this tutorial, you can check out some of the other tutorials in this series to learn about more of the open resources from the Allen Institute for Brain Science. They're all available on our YouTube channel at youtube.com/Allen Institute. So again, a reminder, please put your questions in the Q&A and chat with each other in the chat. And I'm now going to hand it over to Sharmi.
All right. Hello, everybody, and thank you for attending this tutorial. We'll be showing you, my colleagues Casey, Layla, and I will be showing you how to get your hands on that wonderful data set that many of you may have heard about in the main symposium. Before we get started, I'm just going to point you to the repo, the GitHub repo that we have set up for this tutorial. Hopefully you can see my screen being shared right now, we will share the link for this in the chat. It's github.com, Allen Institute, OFS, EM Connectomics. So if you'd like to follow us along the second half of this tutorial with the Python notebooks that we've set up, please go ahead and feel free to take a look at this and set up the installation with these couple of lines, which will just download a bunch of libraries and download some of the files that you need for this tutorial. Okay, so with that, I'm just going to do a short presentation first before we dive in. Okay, so I'm Sharmishta Seshamani, and I'm a scientist with the EM Connectomics group at the Allen Institute. And today I'm presenting with my colleagues Casey and Layla to show you the MICrONS dataset that we released last year and show you some of the tools for interacting with the data and extracting information and do some fun analysis with it. So first, I'll start with a quick recap of just electron microscopy for anyone who may have missed the symposium earlier today. The MICrONS dataset that we're presenting contains data obtained mainly with electron microscopy, which is a modality capable of imaging tissue volumes at nanometer resolutions. So this can help reveal details of all structures within a tissue that are not really possible with many other modalities. So for example, you can see the details of the axons in the mouse cortex, which are on the order of less than nanometers, and synapses and spines and organelles all packed in with all other structures. So here's a video showing you how the electron microscopy can be used to extract biological structures, which can then be further analyzed to study connectivity and morphology. And mapping these types of networks of neurons at the level of these synaptic connections is really the idea behind the field of connectomics, which began in the 1970s with a study of nervous systems of small systems like worms and so on. And modern connectomics really has moved on to mapping more complex organisms, presenting larger data sets and new challenges with that. So the data set that we're presenting today is a mouse data set and is a result of the consortium of the Allen Institute with Princeton University and Baylor College of Medicine, funded by the IARPA MICrONS program. The goal of the program is to advance neuroscience by providing insights for next generation machine learning algorithms. And more specifically, the aim was to map cortical connectivity with electron microscopy, as well as functional imaging, which Nick showed you a little bit about. However, so far we have only released the EM data publicly, so we will be discussing only that in the rest of this tutorial. So the overall pipeline that was developed involves three parts: data collection, post-processing to actually turn it into a coherent volume, and then information extraction to start doing different types of analysis. And we have a lot of this information put out there, and we'll show you how to get to it. So for those of you who attended the symposium, you also heard about some of the really cool analyses that are ongoing. And I just wanted to point you here to some of the publications that we have out there. But please keep in mind that these are just some examples. And what we really want you to think about is all the other cool things that you can think of analyzing with this data set. So although we talked about cell types and specific like mitochondria, for example, there are a lot of other things that you can do. So, detect and study other types of organelles, for example. The Synapse database that's released has a lot of information about the automatically detected synapses and can really provide for lots of different types of analysis. And finally, I'd like to also mention, as Forrest did, that there are other types of, it's not just neurons and cells in there, but there's all the structures that you really see. So for example, vasculature, and if you're interested in doing the studies of vasculature or their relationships with neurons and so on, there's definitely lots of possibilities with this type of data. So finally, I'd just like to quickly thank the entire multidisciplinary team from all the different institutions that have contributed to this data set, and also to our funding agency, and of course to Paul Allen for all his support, vision, and encouragement. So with that, I'd like to point you to just brainmap.org, which is from where you can find links for our data set. And this will take you to the MICrONS Explorer website, which is located at micronsexplorer.org. Just a quick recap that this data set is 250 by 140 by 90 microns of mostly layer 2-3 of the mouse cortex, and it contains about 2,000 sections and about 3 million synapses. So let's now go and see how we can actually get our hands on it. So let's stop that and take you to, if you'd like to follow me, start with the brainmap.org website. So here I am going to scroll down and click on connectivity matrices, which then opens up this page, and under ultrastructural connectomics, the MICrONS Explorer website will take you to the MICrONS, the main MICrONS Explorer location. So to access the data, we're going to click on Data and Layer 2-3. And scroll down where it lists the different releases of the data. So far, we have only one release, but in order to actually view this data set, you can click on this link here where it says data set available at this link. And if you're using Chrome or Firefox, it should easily open up in a new tab, which looks like this. So this is a view of a tool called Neuroglancer, which was developed by Google, which is a WebGL viewer for volumetric data. And this is the main tool that we use for looking through these data sets. And the first part of this tutorial is going to involve actually using this viewer in order to parse through this data and do some basic analysis with this. So let's first familiarize ourselves with what we're looking at here. On the left, we are looking at a 2D view of the actual EM section that was imaged. And on the right, we're looking at a 3D view of some of the objects that are present in it. In the 2D view here, we can see the section that, just to orient you a little bit, the top of the section is towards the PIA, and it's mostly spanning layer 2-3 in this data set. And I'd just like to point out that you have scale bars here at the bottom, which give you a sense of where you are out there. Okay, so the first thing you notice is that there are a couple of highlighted objects here, both in the 2D, which show up in the 3D as well. So the 2D is kind of the cross section, and the 3D is showing you actually meshes of those objects. So the first thing we're going to try to do is a fun thing is to control the colors of these objects. Right now, they're randomly assigned colors. You can hit the L key on your keyboard, which will then randomly assign different colors to the cells. So you can definitely use that to change the colors if you don't like them or improve your visibility. Okay, so now coming back to the 2D view here, the first thing to notice is you have these crosshairs over here, the green and red lines, which really tell you the center point of focus of where your image or your location is. And if you want to go take a closer look towards that that crosshair location, you can press Control and scroll with your mouse, and that will help you zoom in. If you scroll down, and if you scroll up, it will zoom out. So pretty intuitive there. If you don't have a mouse, you can use the plus and minus keys as well with the Control key hit in parallel. So then if I want to change locations of the crosshairs, I can right click anywhere in the 2D image. So if I go ahead and right click, I notice that I can change the location of the crosshair and what I'm inspecting. Notice that the 3D view is synced with the 2D view. And so as I change the location of the crosshair in the 2D view, it's also changing with the 3D view. I can also just simply click and drag with the left click and drag around in the 2D view in order to see, to inspect and change the location of the crosshair. Okay, so now suppose I want to take a look at what some of the adjacent sections are. So as I mentioned before, this is just one section. I can just go ahead and scroll with my mouse, and that will show me what some of the adjacent sections look like. Okay. I'm going to zoom in a little more. You can take a closer look at things this way. Okay, so the other view, so this view shows you just the XY view, and if you hit this icon here, which says switch to 4D panel layout, that should bring up 4D panel here, where again you have the XY section up here, the 3D view at the bottom, and you have an XZ and YZ view here, showing you really those adjacent sections and how the point is oriented in all these different views. So just an easy way of knowing which way the orientation or what the axes are is neuroglancer has colors assigned. So red is X, green is Y, and blue is Z. So it's really easy to tell which view you're looking at what time. So to switch back to the previous view that we have, we can click on this 2D to switch to XY 3D layout again, and that should bring us right back to where we were. And okay, so now we can, so that gives you a couple of examples of how you can navigate within just the 2D or the EM imagery on the left. So looking now at the panel on the right, which is your 3D view, again, you can right click anywhere in this view to go ahead and change the location of your crosshairs. Clicking and dragging, and of course you can zoom in and zoom out again by pressing Control and scrolling with your mouse. Hopefully that's a lot of fun, and as you get to see some of the details, and hopefully these are familiar pictures for those of you who have seen, who attended the symposium this morning. So you can click and drag, and that will help you inspect, rotate and inspect the objects as well. Okay, so now let's take a look at understanding what else we're seeing on this screen. So on the top left here is the resolution information. Over here it says 4 by 4 by 40, which means that each of all the images here are 4 by 4 nanometers, that the pixels are 4 by 4 nanometers, and the thickness of the section is 40 nanometers. To the right of that, we have the voxel location information about where your crosshair is. So note that this is in voxels and not in nanometers. And another way of changing your location of your crosshairs is by simply changing these values. So for example, I can move from 768 to, let's say, 800 by just typing that in, and that takes me to a different section. You can also change XY coordinates in a similar way to navigate yourself around there if you want. Below that, we have some really important information here, which are the names of what we call layers. So the layers actually are what contain the image and segment information, all the information that you're actually viewing on your screen. So the first layer that we have is the EM, is called the EM layer, and it contains the image information. So to make it clear what exactly I'm talking about, I'm going to turn it off. So one way to turn off a view of a layer is by actually just left clicking on it. So you see the imagery immediately turned off, the 2D, the EM imagery turned off and on. And you can toggle views for this by just clicking on it. On the right, we have the segmentation layer, and this contains the segmentation mask, the segmentation information, as well as the 3D meshes. So again, I'm going to toggle on and off, and you see what gets turned off when the segmentation layer gets turned off. So as you saw in some of the talks, really navigating and changing options on these layers and the way you visualize them can help you investigate and analyze the data much better. So in order to do that, Neuroglancer provides us a tools menu, which you can access by right-clicking on the layer name of interest. So in this case, I'm going to right-click on the segmentation layer here. Okay, and that brings up the tools menu for the segmentation layer on the right. So this tools menu contains tabs for rendering, annotations, and transform. But what we're really interested in is controlling visualization and the rendering here. So that's the one that we're going to focus on for now. So the rendering panel has a 2D visualization panel right on the top, which helps you control some of the options on visualizing the 2D view. So in this case, we're controlling the segmentation layer. And if I change the opacity on this, notice the change in appearance of opacity in the 2D view. And similarly, you can just turn it on or off by toggling this other bar over here. Saturation again controls the way you can see some of the color and information on this view. Below that, we have a 3D visualization view panel. And again, opacity allows us to control how we can look at these cells. So if you remember some of the slides from Joanne's talk earlier today, she showed you how this helped her really analyze some of the phagocytosis in her cells. Okay, and below that we have toggling options for visualizing, actually loading the meshes or skeletons. In this case, we don't have skeletons loaded for this data set, so that's not really an option. But if you if you have them, or you want to turn the meshes on and off, you can definitely turn this off and reload the data, and you'll see that the meshes disappear. Okay, below that is a very important panel, which is the segment selection panel. And this really shows you the information about the objects that are highlighted. So I'm just going to zoom out a little bit on the 3D view so that we can see all our objects again. So looking at this a little more carefully, the bottom part of this panel shows you, actually lists each of the objects that you have selected. And the colors here of these rows correspond to the actual object. So the other cool thing is that you can actually go and highlight one of these objects, and you notice that the corresponding row here also starts blinking. So you'll easily be able to identify which object that is. So for each row, you have an object ID, which is unique to that object. To the right of that, you have an option for actually copying that ID. So that makes it easier for you to save it or use it for your analyses. To the right of that, we have a manual color changing option, which is pretty easy and straightforward to use. And to the right of that is a toggle on and off button. So turning on or off this button just lets you keep an object within view. Okay, so above each of these, we have collective options. And the collective options basically apply to all the objects here. So on the right, we have a on and off check box, which will keep things within view or not. And then you have a copy visible segment IDs, which will only copy the IDs that have these toggle buttons on. And then you have a copy all segment IDs option. So I'm going to go ahead and click on that. And to the left of that, you have a remove all segment IDs option. So that's just an X, pretty intuitive. So if I take those off, you immediately notice that all the objects are in the 3D view are gone, and the specificity of those objects can't be seen in the 2D view either. So to put them back on, you can easily just type in the IDs. So since we copied them, I'm going to paste them back. Okay, that didn't work, so I'm just going to go copy those again from another link. Bear with me for a second. Okay, I'm going to go back and copy those IDs. And I'm going to paste them back in the empty one and hit enter. And there you go, your objects are back with the settings that you have. Okay, so this shows you how you can actually select an ID, select an object if you know its ID. But most of the time, you may not know what its ID is unless you've actually seen that object before and already found it interesting. So another easy way and common way that most of our scientists actually use for exploring the data is by actually looking within the 2D data set and saying, oh, here is a cell. Okay, this is a nucleus of a cell, and I want to see what it looks like. So what you can do is just go and in the 2D view, I hope you can see my cursor here, and just double click on it. And that will immediately load up the 3D object in the 3D view. So as you can see, I'm very slowly loading an object there, and you can see now where it is and try to do some analysis with it. So to show you a quick example of some of the things that some of our scientists were doing before, I have another tab open here where I have a pyramidal cell selected. And I have my crosshairs actually on a specific spine. And let's say I'm interested in looking at this spine. And so I zoom in. And now I say, oh, okay. I wonder what its presynaptic partner is. And so I can go ahead and look at the EM on the left. Since the crosshairs are synced, I know exactly what that point is. And so if I zoom in a little bit, I can see that, okay, this purple object is the object that I'm looking at. And there seem to be some vesicles here, and that's probably its presynaptic partner. So I can go ahead and double click on that, and immediately the object of the cell gets loaded up in the 3D view. If I then go back here and I zoom out, control and scrolling my mouse, I can then see what cell it's actually connected with. So if you remember some of the analyses that Agnes showed you this morning, a lot of it was done in this manner. And there's a lot that you can also think of doing in this kind of format. Okay, so before I wind up this section, I just want to point you to a few of the tools on the top right over here. So the extreme right icons are some very self-explanatory icons, which I won't get into. It's about feedback, what's new, help about some of the quick keyboard controls that you can do, and setting your preferences. But the ones I'm really going to talk about are this share button here and the curly braces for editing the state. So the share button is a very, very useful tool in order to be able to save your work or share your work and say, suppose you say, oh, hey, here's a cool spine that I'm looking at, and you have this view of it and you want to save it for yourself or share it with someone. You can go ahead and just click the share button, and that will give you a link that you can copy and save for yourself, so that next time you click on that link, it will come back to exactly this view with the zoom level and all the parameters set exactly like this, and you can continue your work or share it with someone else. And so what it's really doing is neuroglancer is saving what we call a state, which is essentially just a set of parameters that define this particular view. And this information some of this information can be seen in the JSON information, which you can get by clicking on these curly braces here. And I'm not going to go over every single thing here, but I want to point out simply that you have the layer information here together with a bunch of parameters for viewing. And here you can see within the layers that you have 2 layers, and I'm just going to open those up, and you see there's the EM image layer and the segmentation layer, and each of these layers has a source and a type and a name and a bunch of other parameters that are specific to the type. I particularly want to point out the source, which is very useful and will come up in a lot of our notebooks as we are trying to access the data, the image data or the mesh data for doing any kinds of downloads or analysis that we want to do. And this information, I just wanted to point out, is easily available from the neuroglancer link, and you can always use that as a reference. We also have links to this and this information posted on the GitHub page in the cheat sheet, but I just wanted to point that out here as well. Okay, so I've now gone over how you can actually access a lot of the data that we have put out there for you. But very often with your analysis, you may want to put down your annotations or and really keep record of a lot of those things and interact with it in different ways. So I'm now going to pass it on to Casey, who will talk a little bit more about that. Right. Thank you. Casey, it's all you.
Hi. All right. Hi, my name is Casey Schneider-Maisel. I'm a scientist at the Institute, and I work on data analysis of EM Connectomics. Let me share my screen, and Sharmi just showed you how to look at the pre-existing segmentation and imagery data, but often one wants to add annotations. This is a generic term that we use to refer to any kind of extra data that we put onto this. That can be synapses, that could be the location of a neuron of interest, it could be location of vasculature, as somebody in the Q&A just asked about. So there's 2 general categories of annotations that you can think of. One are annotations that you add more or less either manually or perhaps automatically. And then there's another category of annotations that we've already produced for you, which are things like synapses and cell body locations. So since we're already in neuroglancer, let's first look at how you can add annotations in this view. Now, Sharmi showed you before these layers, the imagery layer and the segmentation layer, but you see this plus button to the right. So if you hold either, I think, Control on Linux or Windows or Command on a Mac, and you click this plus button, you'll see a new layer pop up. It's by default just called annotation. And if I right-click that, I get a new kind of widget here over a new panel over on the right. And this panel lets me add different kinds of annotations. The first, you can choose which kind of annotations via this collection of tools down here. The first one, the one that just looks like a little dot, is the one we're going to click on first. This just produces point annotations. So I'm going to click this. You see it becomes highlighted with a little green gradient. And now if I look around my data, either in the imagery, maybe I think, oh, that's a nice looking mitochondria, or maybe, hey, that's a nice looking mitochondria as well. Or if I go into the 3D view and I say, whoa, that's an interesting, I don't know, here's the cilia of this neuron here. I right clicked. You get you can also click on an object in the 3D view. And so this, you see, starts listing up these points in space. I can now click on them to jump straight to them, or I can use these square brackets to move through them as a list. And this lets one start building up a list of kind of point annotations. Now, you might want to know more than just the location in space. You might also want to know what segment ID these have, and you can get that by changing one extra option on this annotation tool, which says linked segmentation, and you bring that and you have that point at the segmentation layer. So now, if I click on the same object here, this new annotation, this fourth one, also comes back with a root ID associated with it, whereas these other ones you see don't. You'll also note that annotations have a description field that I can use to just write in some free text. All right, so this is one way to start annotating the data, and there's other kinds of annotations available too. There's the second one is for line annotations, so that helps you span 2 points. And here, let me make the view more transparent, the 3D view more transparent, so you can see how that looks. So this line annotation spans multiple points. For example, we used that in the chandelier paper to write out, by the way, note that the, sorry, you have to control click to lay down annotations in either the imagery or the 3D views. So we would use this, for example, to identify the AIS of cells. Then there's also a bounding box annotation, a sphere annotation, which is kind of fun if you want to add 3D balls to your space, and some other kinds of grouped categories that I'm not going to go into right now, but you should feel free to explore. So you can also add multiple annotation layers by just continuing to press this button, this plus button, and you can change the name just by typing in here, my annotations, and then you see that changes this layer name. So this is a process through which you can go through. And now I've only done sort of toy annotations, but maybe you are interested in identifying particular organelles in a nucleus, or you want to find specific spines to study the shape of. You could then go to, okay, here's my new spine annotation. Spines, go here, back to this dot tool, and click on a bunch of spines that you think are interesting and you want to measure the shape of. How do you save this or get this out? Well, one way to save the state of what you're doing is always to go to this share button and hit copy in this long link. And now if I go to a new tab and I paste in that link, it will jump straight to this exact state, just like Sharmi showed. However, that's not the easiest for analysis purposes. For analysis, you often want to just export the data to run in some other tool. And for that, there's this export to CSV button, which just does what it says. It downloads a CSV file containing the point location, as well as if you have line annotations and such other information about the data that you've just clicked on. So now I'm going to go and actually ask, well, or help show you guys an example of what you can do with these kinds of annotations, as well as the annotations that we've produced in generating this data set and what we have available on MICrONS Explorer. So for starters, if you haven't already downloaded the notebooks, if you want to, this is the time to do it. So again, go to this GitHub page, go and download this main directory, run this download.sh command after making a Conda environment in this directory, and then start Jupyter. I'm going to assume that you've done all of that and that you have your Jupyter, or sorry, the directory from this repo here at OFS EM Connectomics. I'm running Jupyter Lab. If you were running Jupyter Notebooks, this should look a little different, but be basically the same. All right, so now go in this OFS EM Connectomics, go to Notebooks, and then go to this notebook number 1, synapses, soma, and data. All right. So I've just showed you how to make annotations. So now we're going to import some annotations. The first thing, this being a Jupyter notebook, is in Python, we want to import some packages. This is actually going to be a very light set of packages. Aside from some things we've made, we're just going to use pandas and NumPy. So let's import pandas and NumPy in the standard way. Pandas is PD, NumPy is NP. I've already made an example manual annotation file and put it in the data folder here. So I'm just going to import it in the standard pandas read CSV. And if we look at this, this CSV, it looks pretty much normal, like any other CSV. It has some columns, coordinate 1, coordinate 2, the ellipsoid dimension. So coordinate 2 would be present if we were doing line annotations, for example. Ellipsoid dimensions would be present if you were using sphere annotations. You see segment IDs, that's the ID in that linked segmentation column. And you see, for example, the description tag, which would be the information in that free text field. Now, the one thing that's a little tricky about these CSVs is that the coordinate values are three-dimensional points. And so actually, the pandas doesn't interpret this as a list or a point, it interprets it as a string. And so if I look at one of these, you see, it says, oh, hey, this is a string with 3 different numbers in parentheses. So the thing you'll always need to do if you're going about this approach is convert the string into a list of numbers. Here's one way to do it, is you can just define a function that takes the string from the second character to the second to last character, and then splits it and passes it to as numbers. There's a bunch of other ways to do the same thing. But if I apply this function to the coordinate 1 values now, instead of being a string, I get a new column that is the same location in space as a number. This is a thing that's going to come up a few times here. We find it convenient to have 3D points as data in columns, but Pandas doesn't always agree. Now, it's certainly not sufficient to just look at the data that you've clicked on or made. So we've actually made a bunch of data available to you on MICrONS Explorer. So a lot of this is already either downloaded or on the already in the repo. And I want to start out by just saying how you could download it to begin with by going to micronsexplorer.org, going to data, layer 2.3, and then scrolling down, and you'll see a set of tables here. The synapse table is the table of 3.2 million rows of the synapses between objects in the volume. A sum valence table, the location of most of the soma, the fixed meshes of cells that you'll hear a little bit more about, and a little bit more data. Let's go through some of these data sources individually. So the first and most useful one, well, not first and most useful, the first one that's useful for guiding one's kind of thinking about what is in the volume is the soma and cell types table. So this table has basic information about the center location and general cell type, whether excitatory, inhibitory, or glia, of all cells in the volume. And this can be used to look up specific neurons by cell type or to provide the relative location of their positions in the data set to the cell body, the target cell body. So we can import it just like so with the read CSV. And what each row of this looks like is you have an annotation ID. This is sort of just bookkeeping. You have a cell type. It's an E for excitatory, an I for inhibitory, and a G for glia. You have this point position column, which says where the center point is. And this, again, has this kind of string property that we need to take care of. And this is in voxels, by the way. You have the root ID of the object, and then you have the X, Y, and Z positions in nanometer space. So already having converted from voxels to Euclidean space. All right, this description here just says the same thing. So for convenience, let's turn that point position into a numeric list. And we're going to play the same game as before, where we're just going to split the string like so. And now soma df point position looks like a list instead of like strings. Good. So now we can actually use it for plotting or to get things into neuroglancer. So how do these values relate to what you see in neuroglancer? Let's just pick a random one of these cells. I picked number 10. So we can, one kind of manual way, it's slow, but it's nice for just kind of super exploratory stuff, is to just copy the point position location like so. So it has the 3 values comma separated. And you can actually just paste that straight into the XYZ position up in neuroglancer just by clicking. And now, as I wait for the imagery to load, you see, okay, the imagery indeed shows up right in the middle of a cell body. And just like, I think this is an excitatory cell. And as you can start seeing it, the cell load, indeed, the dendrites are spiny, consistent with being an excitatory pyramidal cell. Great. Sorry, my Zoom thing is right in the middle of where I need to get my tab back. Okay. And these consistent root IDs across all of the tables in neuroglancer and in the data sets we provide are basically how you'll organize data. Now, the biggest table by far is the synapse table. It's about 380 odd megs. And it has a row for each of the 3.2 million synapses found via convolutional neural networks and some other deep learning done by Nick Turner and co at Princeton. The data in this table is mostly right, has approximately 90% precision and recall, but has not been explicitly proofread. So as with the output of any ML pipeline, there are going to be some false positives and false negatives. And if you run across something extremely surprising, it's valuable to double check the raw data if you don't need to look at thousands of instances. So we can import the data just like so. This is one of the files that you might need to run the, that you do need to run the download.sh for to retrieve. All right, so now let's look at what's in each column. Again, you have a bookkeeping ID for that synapse. You have the pre-ID, root ID, and the post-root ID. So the synapse has this root ID as the presynaptic side and this root ID as the postsynaptic side. You have cleft voxels, which is a measure of the size of the synapse, basically the number of voxels in the synaptic cleft. So larger values are larger synapses. And then you have a number of position values. So center point is a point in the cleft itself. Pre-point is a point on the presynaptic neuron. Post-point is a point in the postsynaptic neuron. And you'll see that all of these have VX or NM suffixes, which indicates whether that is a value in voxels or in nanometers. So how do these values relate to what you see in neuroglancer? Well, I just picked a cell ID from what we saw before. I can query its synapses by using this, this nice feature pandas, the .query method, where I say I want all rows of synapse data frame whose value of the post root ID column is this root ID value. And apparently the cell has about 3,700 synapses, input synapses, postsynaptic sites. And just for simplicity's sake, let's look at one of these synapses. So here's that one synapse. I've stripped out the center point, post point, and pre-point values. So let's just look at the center point value for this synapse. Again, I'm just going to copy it here and paste it back into neuroglancer. And let's see if this agrees. Whoops. With being a synapse, and indeed it does, right? This center point lands exactly on this middle ground between this middle point on the membrane between this axon here, which you can kind of tell from seeing all these vesicles, and this spine here, which you can tell because it has a clear cytoplasm. And indeed, we can go into the 3D view and see quite clearly that this is exactly landing right on the face of a synapse. So that's great. And then there's some additional data. There's the proofread soma subgraph synapses. This is a subset of the synapse table only for synapses between excitatory neurons. This has been extremely carefully proofread and also has an additional data point, which is the spine head volume. Otherwise, it's very similar to the synapse table, and we're not going to go into it here. All right, so how do we more programmatically get this data into neuroglancer? Let's start by digging into inhibitory neurons a little bit. So we can use this query function method again to look at, okay, well, there's apparently this is the table of inhibitory neurons, or neurons whose cell type is I. How big is that? There's about 32 cells. There is exactly 32 cells. So let's use a state builder object, which is a package that we've made here, to generate custom neuroglancer links based on pandas data frames. So the idea behind the state builder, which is one of the objects in this NGL UI package that you can get from PyPy and should have been installed if you ran the Conda environment create already, is that you specify each one of these layers in sequence, and some of these layer specifications include rules about mapping columns of a data frame to what you see in neuroglancer. All right, so here we have to specify the image path and segmentation path. This you could have gotten from either the neuroglancer state like Sharmi showed, or it's visible in the README on the GitHub repo page. So for simple states, it's a state builder. After importing it, you just say an image layer config and give it the image path. Segmentation layer config, give it the segmentation path. So then these image and seg objects contain information about how to build those layers. And I can put them together by making a state builder and passing it a list of these layer configs. So now if I just render state, so the state builder object has a bunch of logic in it to render out the layer states. If I just say render state and don't pass it any data, well, that's okay, because nothing that I've configured here is data driven. So if I click this neuroglancer link, you'll go straight into the neuroglancer state exactly for this data set. So that's great, but it's kind of boring because we haven't put data into it. So now let's add data. The idea here is that we make mappings between data and annotations. So we are going to make a mapping between soma points and annotation points, those point annotations that I showed before. So because we're going to do annotation points, we're going to make a point mapper. And that point mapper is going to work like so. So a state builder point mapper, the main argument is the column of the data frame that you're going to pass it. In this case, point position is the column of the soma data frame that has the location of the center of the cell. So we're going to give that point position value here. And we actually would like also to bring the root ID along, so it's easy to look up which cell is which. So we add a linked segmentation column, and we give that the column name of the column that contains the segmentation ID, which in this case is point root ID. So now we build an annotation layer that uses this mapping. So the annotation layer config, just like image and segmentation layer config, first it has a name, and then it has mapping rules. And so we give it the name soma points. We assign mapping rules to points, and we make a linked segmentation layer, and we just say, hey, that's the segmentation layer that we've already defined. It's the name of it, so it's seg.name. And now when we build our state builder, we pass all three of these layer configs, the image layer config, the seg layer config, and now this new annotation layer to our new state builder. So run here. And now when I render the state, I don't just give it the return as HTML to make my link. I also pass it a data frame. And what this is going to do then is it's going to take this filtered data frame, and it's going to make a point for every row of the data frame with the information I provided. So let's click this link here, and you can see now I've automatically generated this kind of constellation of points. And if I click on one of those points in the annotation column, and I hit square bracket to move between them, and then I wait. I'm actually being a little slow, but I can tell on my computer I'm downloading a bunch of information. Then as I walk through these annotations, it will be able to pull up, here we are, it will start pulling up the mesh of each one of these objects. And I can use the square brackets or just click around to navigate through all of these different neurons. So that's kind of the basic concept behind using the state builders to bring data frames into neuroglancer. Let's go back here. So let's look at one other example very quickly before I pass this along. So how do we visualize a connection? Well, we can use the synapse table. So let's ask let's look at synapses from inhibitory neurons to excitatory neurons. So we can play the same filtering game to get a list of inhibitory soma IDs and excitatory soma IDs, and then use this query to generate a synapse data frame that's filtered only to synapses between excitatory neurons in the data set and from inhibitory neurons to excitatory neurons. And now we need to turn this list into connections between specific neurons. So we can group the synapses in this, just using kind of standard pandas. We group the synapses in this data frame by common both pre and post root IDs, because a connection can have multiple synapses. So then we can count. You can use this transform function with count to give every synapse an additional column, which is how many synapses are between the cells that belong to that synapse. So this syn in con, let's actually just quickly look at this. syndf head. So now I have an additional column in this data frame that is how many overall synapses are part of the same connection as this synapse. All right, and we can use the pandas describe function to look at the distribution of these, and wow, there's one connection in here that has 21 synapses. What does that look like in the data? So we can play the same mapping game. Now we have to make a new column, center point position, and we can now have two linked segmentation columns for both pre and post root ID. We build the same state builder with exactly the same logic, and we can now pass it, hey, show me the connection, show me the synapses for the connection that has 21 synapses in it. And now these are these synapses. We can now bring up the neuron involved. Here I'm going to make us transparent by making the 3D opacity a little light. And we can see this red inhibitory cell mesh coming in and all of its beautiful synapses onto this green excitatory cell. And as I think you can start seeing, it really looks like this cell is laying a lot of interesting synapses down on the apical of this target neuron, although not entirely. So this is what I've shown you here is how to get data from the tables we've provided and manual annotations into neuroglancer for quick visualization. But you might also want to be using the meshes as a source of data yourself and exploring the meshes on your own computer. And so for that, we're going to pass things off to Layla, who's going to continue telling you how to work with this data.
Hi, everyone. I'm Layla. I'm a grad student at the University of Washington and have been working with the EM group at the Allen Institute for a couple of years now. So like Casey said, I'll take you through how to work with meshes programmatically and figure out how to analyze and visualize them yourself. So to do that, I will share my screen. Okay, so can everyone see the GitHub repo?
Yep, we can see it.
Great. So if we're starting back here, if you want to scroll down, hopefully by now you've downloaded the necessary files. We're going to be working off of Notebook 2 here for mesh analysis. But before we do so, I just want to point you to a couple extra files that were too big for GitHub. So if you scroll down here, you see example meshes for tutorials. If you click on this, it should open this Figshare file folder that I've just put the two meshes that you need. So if you click on the data set here, you should be able to download these two meshes. And the name is really weird because it's the unique object ID, which we will call in just a second. And so to start off again, let's go back to neuroglancer. And in all honesty, I just went around and started clicking until I found two pyramidal cells that I liked. And so you can then go through and copy the IDs by clicking on the two clipboards. And so if you would like to follow along, but with your own special cells, feel free to do that. Otherwise, please download those two meshes. So if we're starting here at the notebook, again, the goal of this section is to show you how to load meshes, and there's two different ways to do that. We'll then look at combining that with the synapse information and some of the synapse tables that Casey was previously talking about, and how to map synapses onto your mesh. And then we'll go through a couple ways to visualize your cells, both in 2D and 3D. And once you have a mesh loaded with your synapses mapped onto that mesh, you can then use graph algorithms to start doing different types of analyses. And as a prerequisite, make sure if you use the Conda environment that we had set up, you should already have Mesh Party. If not, please make sure to install that. And again, you can link to the two example meshes here. And instead of using the file with all of the synapses, we'll be using the SOMA subgraph synapses, which is just a subset. So it's a smaller file. You should already have it in your data folder. And those are all of the synapses between excitatory cells that have their somas within our layer 2-3 volume. So we'll start out by just importing a couple necessary packages. The only unique one here is Mesh Party. And then, as I mentioned, there are two ways of which you can load in a mesh. The first is if you already have your mesh pre-downloaded. So if you have downloaded those meshes off of Figshare, what you want to make sure you do is tell me where the data is. So make sure you've defined your data directory to the proper, to wherever you have it downloaded. And the second way, which I'll show in just a second, is you can directly download any mesh off of from Neuroglancer if you have the segment ID and through using Cloud Volume. And so Cloud Volume is a package that was developed by our collaborators in Princeton, and it just allows you to read and write from the data that you see on Neuroglancer. And Sharmi will go into how to do that in the next section. So here, the segmentation source is how we access the 3D segmentation data. I have it predefined for you. If you don't have this, you can go to the GitHub page or on your Neuroglancer link. It is held within that JSON. And what you want to do is define this mesh meta object, which just allows you to encompass the cloud volume information. So we'll run this. This is just defining points for the data so I can access it. And now that I have already predefined my segment ID, but again, if you've chosen a cell of interest, you just want to change this so that you point to your unique object ID. And the first way I'm going to show you how to load in the mesh is right off of disk. So I want to make sure that this mesh directory is where your data is. And then you can just put in mm.mesh with your mesh file here, and that should read right off of disk. And so that's usually quite fast. If you want to make sure that you actually do have a mesh here, you can look at the number of vertices and the number of faces of this object. So these are non-zero. It means I actually have data stored. So this is the first way of which you can load in a mesh. Let's say you don't have the mesh pre-downloaded. All you need is to have the segmentation source, which I had defined up above, and your unique segmentation ID. So here I've got a second ID. And for the second mesh, rather than reading it off of disk, I'm going to download it directly from Cloud Volume by running this here. And so to make sure, again, that this worked, I have a non-zero amount of vertices, number of vertices in my second mesh. So here I have two excitatory cells loaded into my notebook. And so now I can start working with them. But let's say you want to figure out, well, where are all of the synapses onto this mesh? The first thing you need to do is read in the SOMA subgraph file. So again, this should be in your data directory. You can also use the very large synapse file if you would like to look beyond this cell of interest. So we're reading in the SOMA subgraph, and just to give another overview, as Casey went through, what this data frame now has, it has your pre-root ID, your post-root ID. It has the actual location in both nanometers and voxels. And what's unique about this table is it also has spine volume, which is not the case for the large over-encompassing synapse table. So I have all of the synapses here between all excitatory cells with somas in the volume, but I'm only interested in those that are synapsing onto my cell of interest. And so here, all I'm doing is filtering out this large soma subgraph for the postsynaptic cells on my postsynaptic synapses of the cell of interest. And this synapse XYZ is just creating an array with the XYZ positions in nanometers. So if we run that. So now I have the mesh vertices for my cell of interest. I also have all of the synapses onto this particular cell. Let's create a 2D visualization just to look at it and make sure that it makes sense. So here I'm just using simple matplotlib scatter plot where I'll be plotting a small point for all of the X and Y positions of my, for each vertex in my mesh. And I'll do the same thing with the synapses so that I can see them here as well. So what I'm looking at here is just a very simple scatter plot, but I can clearly see this is my excitatory cell in blue, and in large purple, obnoxiously large purple dots here, you see where the synapses would lie on the cell. Okay, so if I do want to run, or if you want to run any graph algorithms on your mesh based on the synapses, you want to be able to map the synapses to your mesh. And so to do so, I'm going to filter this a little bit more by taking another subset of this. So I previously loaded in a second mesh. So if we go back to neuroglancer, I have these two excitatory cells, and I want to see all of the synapses between them. So what I'm doing here is I'm taking a further subset where I'm now only taking the synapses if the pre-root ID matches that second cell that I had outlined earlier. So if we run this, this has now made my synapse data frame even smaller, and it will only encompass the synapses between these two cells. And in order to map it to the mesh, I'm going to run kdtree. And so what that means is I'm going to map each synapse to the closest mesh vertex on my cell or on my object. Okay, and so here, this will show all of the distances between the mesh and the synapses. And so you can see there's roughly 5 synapses between these two cells, and they're within 67 nanometers. That's the furthest distance. So they're quite close. And so now let's say you want to run a graph algorithm. You want to see what is the distance or the shortest path between some of these synapses. To do that, we're going to import the sparse package from Sci-Fi, which allows us to do this. And we'll run a simple Dijkstra method here. And what will that will give me is a distance matrix as well as a path matrix. And so this predecessors matrix, in other words, will give me from every point kind of what the path is to this first synapse, which I set kind of as the root here. And so for sake of an example, let's see if we can map the path between the very first synapse and the fourth synapse. And so this here will do that for me. And let's make another 2D visualization. So very simple to the one we made before, except this time I'm going to add a line to delineate that path between the first and fourth synapse between these cells. So the only thing I have added to the code from the one before is mapping the path. And so if we run this here, you can see in dark blue, there's the path between the first and the fourth synapse. And you see that I've changed the coloring. And now that I have the synapse information and where they are on the mesh, I can recolor every vertex on this cell based on this distance from the very first mesh. And so the darker the color here, the closer it is to the first mesh, which makes sense as this happened to be the first synapse here. And so the darker the color, the closer it is to the synapse. And as we get further away, you start getting into the greens and the yellows, and that means it's quite far away from that first synapse. And again, that dark blue is the path that we've been calculated on the mesh. So this is just an example, a simple example of some of the type of analyses you can run on your mesh. Some of the more exciting bits is just visualizing it. And so I'll show you a couple ways to how to visualize these cells in 3D. And so there are two packages. The first one I'll show you is called ITK widgets. This allows you to do really quick and simple visualizations within your notebook. It doesn't give you too much control over the actual visualization, but I tend to use it when I just want to make sure that what I'm doing is correct. And if I'm doing any mesh filtering, that I'm seeing the correct thing. And so here what you want to do is you need to create poly data objects. And so if you have downloaded Mesh Party, a really helpful part of that is TryMesh VTK. And TryMesh VTK has multiple functions in there that will allow you both to get your meshes in the correct type for both ITK widgets and VTK, which I will talk about in just a second. And so here what you're doing is you're converting your mesh to polydata. You're converting the path as well, so your skeleton as well into a polydata format. And then you're defining this viewer. And this viewer is going to take in these polydata objects. So here I have the polydata for my mesh. So this is the cell, and the polydata for the skeleton path. So this is still going to keep that path between the first and the fourth synapse. You can then define different colors and opacities in order to make it a little easier to see. And so if we run this, it takes a little second. Okay, and then we call on the viewer. This should pop up within your Jupyter notebook. And you can see now I have my cell of interest in yellow, and the path, if you just scroll in, as you zoom in, you can see that same path between that first and fourth synapse is in blue. And so this allows you within your Jupyter notebook to just quickly throw this in 3D and double check quality control or just make nice visualizations. So the other package that is also for 3D viewing is called VTK. This is another open source package. And this one I tend to use if I'm trying to make more complicated figures or even making a movie. It gives you a little bit more control over the camera and the types of things that you can visualize. So the only difference here is rather than a polydata object, we've defined it as an actor. So rather than a viewer with polydata objects, VTK will have a camera with actors. And so what we're doing is using TryMesh VTK again. You're going to create a mesh actor from your mesh object. I'm going to put in our second mesh so we can see both excitatory cells again. And now I'm creating a point cloud actor, which is actually going to create a 3D sphere for every synapse between these two cells. Again, you can denote the size. You can change the size. I've made these very large so that we can see them and play around with the colors if you choose. Again, so the camera object here, all it needs is what the start point is. And so I've given it the centroid of my first cell. And you can also decide how far or zoomed in you want this camera to start out. And then lastly, you want to use TryMesh VTK's render actors, which you can pass in as many or as few actors as you want. And since I have created a camera object, I'm also passing that through. So if I run this, one thing to note about VTK is that it will not run within your Jupyter notebook. So it'll come out as a pop-up, a little Python pop-up. And so here you can see my original cell. Oh, let's take a second. Let's try running it again. Okay, there we go. So here now I have a 3D view of my cell of interest here in gray, the second excitatory cell here in blue, and you can see the synapses between these two cells are these spheres. And so if I decide to zoom in on one of them, let's see this guy here, whoops. You can see that this is in fact a synapse point as the intersection of the blue axon and one of the gray spines. And so this, using VTK, allows you to make more complicated visualizations, as well as small movies and videos if you choose as well. Right. So that wraps up our bit on how to use meshes, both for analyzing them and visualizing. I will now pass it off to Sharmi, who will help wrap up the last bit of this tutorial.
Hello, thank you, Leila. Let me share my screen. I'm going to do a very quick tutorial on simply, on one of the last notebooks that we have put together. So again, going back to the GitHub repo, it's in notebooks, and it's the third one, which is downloading images and segmentations. It looks like this. So I'm going to go over this really quickly since we are running a little short on time. However, these notebooks are all up on the GitHub repo, and feel free to please access them and use them. So in this tutorial, I'm going to talk about downloading some of the images and segmentations, just image segmentations. Leila showed you how to actually download the meshes that are generated off of the segmentations. But in the case where you might be interested in actually analyzing some of the raw imagery with the segmentations, for example, for creating your own detector of a specific organelle, or even for just doing some sort of image processing and analysis on the images themselves, you might be interested in actually downloading these from source. So some of the tools, the libraries that you want downloaded are Cloud Volume and the Imagery Client, both of which are installable with pip. Okay, so to start out, we just have some basic imports, pandas, matplotlib, and numpy. And the very first thing we're going to do is to set up, I should run that cell, is to set up the image sources. So just as I had pointed out in the neuroglancer tutorial, you have an image source, the EM imagery source, and a segmentation source, which are two different sources and that have been used, and that Casey and Leila also went over. And so we're going to say, okay, these are the places we want to download our images from. And I'm going to show you actually two methods of doing it. One is by directly using Cloud Volume, which has a lot of the power of directly actually going and grabbing that data, and also a second method, which is a kind of wrapper that's written for actually downloading that to more specifically for our data. Okay, so the very first method I'm going to do is directly with Cloud Volume, and here we first start out with the import statement, and we say, okay, let's first initialize our Cloud Volume connection and say we want to download some of the EM imagery. So the very first thing we do is to set up that Cloud Volume connection with this command, and we say, okay, now that we have these this image source connected, what are the bounds of the data? And if you look at that, it will tell you what the XYZ bounds are. So this is the lower left corner, and that's the upper right corner. So and these are all in voxels. So it tells you really how much you have in there. So the first example we're going to do is say, okay, let's just pick some arbitrary 500 by 500 box in the middle of the volume, and say, let's say one section. So I'm going to just pick some bounds somewhere in between, right in the middle of the volume here, and say, okay, I'm going to define these bounds this way and download the image, which I can do right away with this command. So if I look at the shape of the image that I've downloaded, it's 500 by 500, and it's just one slice here because I'm just going one in Z. And now I can actually look at what I've downloaded simply with matplotlib. And voila, there's the EM imagery right there. Okay, so now I can do something very similar with the segmentation. Again, I initialize the cloud volume connection right there with the segmentation source this time. And if I look at the bounds, notice that these bounds are different from the ones we had before. In particular, if you scroll up your notebook, you'll notice that this is actually exactly 1/2 the size of the image volume. And the reason for this is that the segmentation was actually computed on 1/2 of the resolution. So the original images are 4x4 nanometers, and this is 8x8. So, so that's why you have less pixels or voxels in this particular bounds. However, you can easily relate them by reshaping them in either direction. Okay, but so let's go ahead and now download a piece of that segmentation. Let's just divide the bounds exactly by 1/2 of what we had downloaded before. And we'll say, okay, let's go ahead and that's going to download. And let's say, okay, let's look at it. So we'll run the same command again that we ran for the EM imagery. But unfortunately, what we see is something like this. And the reason is that the IDs, if you remember, so the segmentation really contains the IDs of the objects that are present in that particular box itself. And some of those are just very large values, and there's no way to easily control that range. So in order to actually look at them nicely, you can easily remap these. And we use the fast remap toolbox. You can use the fast remap toolbox. It's you can definitely use just numpy unique and write your own command, but fast remap provides that and does that pretty efficiently. So I'm going to show you how to use fast remap here. So the first command here is just unique, which will actually calculate all the unique IDs within that box and give you the counts. So if we look at those unique IDs, it's now telling me, okay, all the unique IDs inside that box are listed here. And now I can actually remap those and renumber those to something else so that they're not these really weird values all over the place. But let's say I just want to label it from 1 onwards. So I'll say, okay, let's go ahead. And so now I've remapped all my IDs between 0 and 18. So I have only 18 values. And that's stored in this remapped seg variable here. So I can now visualize this pretty easily by just, again, using matplotlib, and that looks something like more like what I expect. So as I mentioned before, the size of the segmentation and the imagery are different. So if we actually look at this, we'll see that the segmentation shape is 250 by 250, whereas the image shape was 500 by 500 for exactly the same thing. So if you want to overlay these things, you definitely have to do some sort of reshaping in one of the directions. So instead, we've developed a cool toolbox called Imagery Client, which automatically does a lot of those things for you. And might be easier for a lot of the purposes. So I'm just going to show you how to do something similar now using this toolbox. So the first thing we're going to do is import Imagery Client. And this again should have gotten installed with the environment if you use the environment YAML file. We initialize the Imagery Client with the same image and segmentation source that we used before. And now let's do something slightly different, not just pick some random box. Let's say we want to do something interesting where we want to go pick a synapse and look at the imagery around it. So I'm going to read from this CSV file, the SOMA subgraph synapses, which Layla just showed you about, where she actually mapped some of those onto a cell. And I'm going to randomly pick some synapse, let's say the 400th one, and grab the postsynaptic cell ID and the presynaptic cell ID for that specific synapse. Okay, and so I run that cell. And now suppose I just want to look at, 5 consecutive sections, like around that synapse, a couple, 2 before and 3 after. I can go ahead and set bounds using the center position of that synapse. And I say, okay, I'm going to go in XY 150, 150, so that I have a 300 by 300 box, and then go 2 Z's before up to 3 Z's later. And so I'm setting my bounds this way. And once you have the bounds set up and the imagery client object initialized, all you need to do is just run this one line of code to go ahead and actually download those cutouts. And what this will do is is to give you 2 things back. One is actually all the imagery, and the second is the segmentation masks. So the imagery is simply just a NumPy array, a 3D array that contains all the imagery information. And this one is, the second object is actually a dictionary, which contains each of the IDs of each of the root IDs or the objects that are present. It has an entry for each of those in the dictionary, and contains masks basically. So it's 1-0 values of those, so that you can actually identify. So for example, if you want to look at the presynaptic cell and the postsynaptic cell that we have identified here, you can specifically look at those rather than all of the objects inside the segmentation. So let's go ahead and run that. And once we have that downloaded now, we can try to plot these. And so what we're doing here is really showing the images, as well, the EM images, as well as the postsynaptic and presynaptic masks in those images. So you can see these are, if you scroll through these, you'll see these are consecutive images. And again, if you look at the 2 columns here for the postsynaptic ID as well as the presynaptic ID, you see how that how the mask changes over consecutive sections. So you can now save these images and perform any kind of analysis that you would like to. The last thing I'd like to point out before we wrap up this section is that the imagery client has actually resized all of these images to the same size, so you can actually overlay these directly and use those masks directly on these images, which can make your life really easy. Okay, so with that, I'm going to just quickly point you back to the repo. We just have a couple of minutes, but I'm just going to say that a lot of information is here. The important links for the tutorial. This will lead you to the neuroglancer view, and the notebooks are all here. We also have a cheat sheet that we have up here for all the data that you can download for this particular data set, the image and segmentation sources, all the tables that you can download from a MICrONS Explorer website or from this little cheat sheet over here, which has links. This last thing really has meshes of like all the neurons that have been analyzed in this, and this is a pretty large file which we didn't download for this tutorial. And finally, there are some additional resources. You can always access the MICrONS Explorer website through the brainmap.org portal. And this is the MICrONS website, of course. And finally, I'd like to say, if you want, there are a lot of additional examples other than what's in this repo at this location. the GitHub MICrONS binder location. So if you're interested in looking at some of the code was used for example, mitochondria work that Nick had presented earlier, you can definitely access it there, along with some of the data that needs to be downloaded for that. So please definitely visit this, and I hope you enjoy using this data set. Thank you. All right.
Thank you, Sharmi, Layla, and Casey for that great tutorial, and also for answering all of those questions in the chat so that people can see all of the responses, excuse me, all of the responses to those questions that they had. We'll post those questions and answers in our community forum and add a link to both the community forum post and to the GitHub repo in the video description on YouTube of this tutorial. And please use that forum post to ask additional questions of our presenters afterwards. As you are working with this, this resource, if you have any questions, please don't hesitate to post them there. So that does bring us to time. Thank you again, and I hope we see you at a future event. Thank you, everyone.